I lead the Sculpting Evolution group, which is a new group at the Media Lab. We just launched a couple of months ago. And we focus on evolution because it is, of course, the reason behind everything that lives. But it doesn't just apply to things that live. It's a law of nature, an emergent law of nature, much like the laws of thermodynamics, that applies whenever you have an informational pattern of any sort that replicates and creates variants that are slightly different. That means that it's not just we ourselves who are shaped by evolution, but also our culture and our sense of who we are, including our morality. The problem is that evolution itself is amoral. It has no moral compass. The fact that we have one is the product of a very rare confluence of particular evolutionary parameters. And so our goal as a group is to fix it. You got to start grand, right? So first of this, just to skate through a couple of projects that are relevant, we want to understand how evolution works. So one way to do this that a particularly brilliant young graduate student who has just joined the group is working on is she said, well, let's take this synthetic ecosystem that Deborah mentioned that we developed previously, and let's use it to evolve parallel populations, thousands of them all at once, just slightly tweaking the different conditions in order to understand how that changes the outcome. Because right now, evolution is a black box. It's tremendously useful. We don't really understand how it works. This is an attempt to open that box. We need to apply what we learn to better the lives of people. And one way we're looking at doing this is controlling the fitness of microbes living inside of us, which we've recently learned play a tremendously powerful role in shaping our own health. If we can control the fitness of these microbes in the gut, then we can engineer them to release therapeutics, resist invasion by pathogens, and generally improve our well-being. But we also need to think about the other organisms that we affect. And in science, I haven't done much work with animal models previously. And now that we're interested in doing some of this in the microbe work, I was confronted with the fact that a lot of animals do suffer in research. And we like to think that it's for a good cause, but it would be better if they didn't. And so one of our other projects attempts to make mice for which we can completely switch off pain perception. No more just using analgesics that are inadequate. No more saying, well, they're the control group. That is what it is. We need something better. We need to reduce suffering closer to home. And if we can do it closer to home in our laboratories, then we can think about moving it perhaps beyond and question how far should we go in reducing suffering in the world, in nature. But the central problem that we're concerned with is that we, as a society, desperately need innovation. We need new innovative technologies because our current way of life is simply not sustainable, resource-wise, environmental stability-wise. You all know that this is the case. The challenge is that increasingly powerful technologies threaten our civilization, our existence, but even more so, our highest ideals. That's a very grand and sweeping claim, but I'm going to give you a very specific example with which I am perhaps too familiar, and that is what is called a gene drive. Now, when you engineer a genome, the way we do it nowadays is we cut it with an enzyme known as CRISPR, or also known as Cas9. You probably heard about it in the news. It makes editing anything about a genome much, much easier because it's essentially a molecular scalpel that we can program to cut and thereby edit any sequence we want. So we normally use CRISPR to cut the target sequence and then introduce a desired change. And this guide RNA molecule tells CRISPR where to cut. But if you put all of these together on one sequence of DNA, then you put them into the cell, CRISPR system cuts the target gene, and the cell incorporates all of it. But what you've done now is you've put the editing machinery itself into the genome. So that is, the cell now knows how to edit itself. And so it's going to express the editing system and cut the other chromosome. So now you have two copies. Since you have 2 copies in this organism, that means that when it mates with, say, a wild-type organism, all of the offspring are guaranteed to inherit a copy. And, of course, each of them now encodes the editing machinery. So in each of the offspring, it's going to cut the other copy. So now all the offspring have 2 copies. And when they mate with wild-type organisms, again, editing occurs, and they have 2 copies, which means down and down through the generations, our initial desired change that we made in that organism in the laboratory is transmitted to eventually the entire population. All that is required is that organisms must be CRISPR amenable. We have to be able to make them transgenic, which is true of a goodly number, but there are many more that we can't for now. They have to reproduce sexually, because again, this only works by distorting inheritance. And they have to reproduce fairly quickly, because it's transmitted down through generations. So if you want to see an effect, then the generation time has to be very short. We could do a tremendous amount of good with this technology. Think altering mosquitoes so they can no longer spread disease. Get rid of malaria, dengue, chikungunya, Zika, yellow fever. Immunizing animal reservoirs of disease. Lyme disease is carried by the white-footed mice that live outside. They're just about everywhere. Aiding threatened species and controlling invasive species. Cane toads in Australia. Did you know that mosquitoes are invasive to Hawaii? Wouldn't it be nice to go to Hawaii and not have to deal with mosquitoes? They're also wiping out these beautiful, endangered native Hawaiian birds, the honeycreepers. If we don't do anything, then in a few decades, they're all gone. Agriculture. Sustainability of agriculture is a big problem, especially given the comparatively fragile state of our ecosystems and the fact that there's ever more humans that we need to feed. Instead of using toxic pesticides and herbicides, how about we alter the pests so that they go about their normal ecological roles, but don't like the taste of our crops? This is, in essence, a way to speak nature's language, because nature operates in the language of DNA, and now we've finally learned to read and write it. But how are these things going to evolve over time? Are they going to spread into related species? These are only some of the many questions that we need to answer before we consider doing anything of the sort. So we're looking to address this by studying the humble worm. And we chose the worm simply because it reproduces very, very quickly, and we can grow a lot of them in the laboratory. There are a tremendous number of organisms out there. That's one of the reasons why natural evolution is so powerful. And if we want to know how something is going to evolve out there, we need to grow a similar number of organisms in the laboratory. And we can grow 1 billion worms in a liter of culture and undergo 100 generations a year. So by propagating drives in this system, we hope to learn how they might evolve and change in the wild. Best of all, this is easy. So we have some young members of the audience in the front row. You could do this. I'm not kidding. We want to put this in schools, in science museums, and generally enable all sorts of citizen scientists of any age to join in this, because this needs to be something broader than just professionals in the laboratory. And the reason we need to do this is because this gene drive example that we came up with here via CRISPR is what we call a global drive system. That is, we must assume that it will spread from just one released or accidentally escaped organism to every population of that species around the world. What would an accidental release of that do to popular confidence in science? Remember, we need new technologies, but at the same time, they're dangerous. It's not theoretical. We've now done it in yeast, fruit flies, and 2 different species of malarial mosquitoes. And in the fruit fly case, we were that close to an accidental release. They didn't know about gene drive when they did this. They developed it for a completely different mechanism. It's a completely different purpose, only for the laboratory. And they encouraged other scientists to use their method, not really understanding the dangers. That close. So we've been working on ways of safeguarding these experiments and got them on board to let others know that people need to take greater precautions. But that's just one aspect of the problem, because the bottom line is that this is something that can be done by a single scientist. A single individual can, in principle, unilaterally alter shared ecosystems, ecosystems that we all depend upon, and that we all have a responsibility to pass on to our children. So what does this mean for our democratic ideals? What does it mean for our autonomy? What does it mean for informed consent? These are all very uncomfortable questions that we face. And our response is, well, first, try to centralize our capabilities. That is, frankly, we don't want everyone to be able to do this whenever they want. It's reversible. It's like a software update for biology. But just because it's reversible doesn't mean you want to have to reverse it every time some young person in their garage decides they want to sign their name in the genome of flies outdoors. So Cody Gilliland, who's a research scientist in my group, is developing a way that he first developed in his PhD of making transgenic organisms very easily. Now you might say, well, that's a problem, right? You don't want to make this too easy so that everyone can do it in their garage. But it's a way that builds a computerized system for microinjection that could work for a lot of different organisms, but it's best centralized. That is, we want everyone to outsource this to a company, because companies can then screen to see if they want to put in a gene drive. But more generally, we need to promote open development of this kind of technology, because no regulation is as good as collective scrutiny, as knowing what is going on and being able to keep an eye on it and make sure that people are using adequate safeguards. And frankly, people have a right to know when other people are running experiments that could affect their lives. Public notification and discussion must always precede and guide experiments with shared impacts. And that's why when we first published on CRISPR gene drive, we described what it could do and necessary safeguards and called for regulatory reform and discussion before we'd done any experiments in the lab. We hope that that will set an example for the rest of the field who are developing some of these applications, because we need a more responsive model of science. We need community guidance and ownership of these technologies that will affect all of us. And we need to somehow foster broadly inclusive discussions and listen and respond to people's suggestions, because scientists are only human. We miss things. We have blind spots. And the more people who take a look at a problem who could potentially identify something that might go wrong, the better. And they need to do it early in the process so that we can redesign the technology as needed. We need a forum for society-wide information sharing and deliberation, which is, again, what our responsive science project is about. We're trying to lead by example. Joanna in our group is developing what I mentioned earlier, how the white-footed mouse is the main reservoir of Lyme disease. Well, we're talking with local communities on whether we should attempt to alter white-footed mice so that they can't spread Lyme or other tick-borne diseases. There's some different ways we could potentially do this. And initially, we would do it certainly without any form of drive system. This could be tested on offshore uninhabited islands, and then potentially eventually on inhabited islands. And if it worked really well, if there were no unexpected ecological effects there, then we could have a discussion about maybe we should consider using a drive system to spread this through the mainland. Because if you take out the primary reservoir, if you make the mice unable to harbor the Lyme bacterium, then the ticks will not be infected when they bite the mice, and then we won't be infected when the ticks bite us. It's a problem that is destroying the iconic image of childhood in America, but it's also one that is not killing children every day, as in malaria, meaning we do have time to do this the right way, to be, as we New Englanders so like to be, the shining city on a hill, providing an example for the rest of the world. And above all, we need to change how we do science, because right now the system is broken. It's inefficient. It incentivizes us to be secret and not tell people what we're doing, and that promotes acrimony. both between scientists and between scientists and the public. And it's dangerous, because even though gene drive is a pretty poor bioweapon, because it's reversible, because it's slow to spread, and because it's easy to detect, you could very well imagine a case where that would be otherwise. And in that parallel universe, it would be my fault. So I'm very concerned with changing the system so that we have better ways of identifying what technological boxes we might be about to open and deciding whether we should open them, because once we do, we're not very good at putting them back. The problem is, this requires a cultural change in how we do science and how we as a society interact with scientists. And that's a cooperation problem. How are we going to bring people together to chart a new course? How are we going to sculpt the evolution of our culture in a better direction? And that's where I'm going to leave it to Tod.
Wow. Oh my, that's that's quite a presentation to follow, as I knew it would be. And I'll make my presentation short so that Kevin and I can discuss, and we can talk a little bit, maybe technically and conceptually, about how music and the arts and biology intersect. But the area I'd like to concentrate on is where Kevin ended, which is the ability of the arts and music in particular to convene people and break through boundaries in quite unusual ways, and probably in radical ways that are somewhat different than we think about music right now. So I want to start just with two quick stories. How many people know who that is? Not that many. Wow. That's Marvin Minsky, who was one of the great MIT people, passed away 2 months ago, almost to the day, and was, as you probably know, one of the people who founded the field of artificial intelligence, spent most of his career trying to understand how to model with computers the way people thought, but also to understand how people thought. And we just had a big celebration here at the Media Lab about a week ago for Marvin. And we were very lucky because Marvin also grew up as a piano prodigy and spent most of his life trying to understand music and trying to use music as a model for how the mind works. And this was really important because I think the first point I want to make is that the arts in general, but music in particular is the one I know the best. It's not just a little pleasant self-enclosed system that we use for entertainment. It's one of the major forms that we use to understand ourselves and to understand our world and to understand each other. And in some ways, it's one of the most powerful forms that we have. And Marvin was fantastic because he spent his career trying to think about music completely out of the box of the way most people think about music. He didn't think about traditional chords or how notes fit together. He tried to figure out why we like it, why people respond to music. Here's just a tiny bit of Marvin. I did a lot of interviews with him. Here's a little interview of Marvin trying to explain something about music.
It's very compelling. When a baby cries, it's very hard to stand there and not go and help it. Is that what's happening in most music? The music making you insist on doing something about something, except it's different from words because you don't know what the something is and you don't know what the something you're supposed to do is.
So it's a funny idea, but it's profound because in that particular case, Marvin hypothesized that music uses our brain to think. In fact, one of his ideas is that music exists as a way to rehearse thinking, where you don't have any negative consequence, so you can try all kinds of radical ways of comparing and making decisions. So it's a rehearsal, it's a way of rehearsing our emotions. And in this case, he says, since it tells stories, but we don't know what the stories are, it's a way of engaging us in filling in those stories in a much more active way than if we're told explicitly. So for Marvin, music was a window into who we are as people, maybe the most profound window we have. We go from Marvin to this image. I went down, how many people have seen this image in the New York Times? It's been all over the place. So you know more about the sheep than about Marvin Minsky. That's interesting. With my class, we went down to New York a few days ago, and 2 days ago saw this performance at the Park Avenue Armory. It's an unclassifiable work, kind of an opera by someone named Louis Andriessen, great Dutch composer, staged by Heiner Goebbels, director-composer. And it was very extraordinary work because it was an opera with no love story, no one got murdered, no political drama. It was about history and physics and the difference between matter and spirit and things which, when the artist gave a talk before the show, they said, we can't really explain this. And we all sat there listening to them saying, yeah, you really can't. We don't know what you're talking about. But turns out they staged this incredible piece. The main actors were a flock of sheep. And so in the fourth act, after about an hour, this orchestra of 60 players, sound all over the place, these sheep come out on stage. And besides it being, so everybody had read about it, there was even an article in the New York Times about the sheep being the most famous operatic divas in New York now, and where they slept and where they found them and what they ate. But when they came out on stage, none of us could have expected what happened. Here were these beings that were, they're living beings. There was no conductor telling them what to do. There was no shepherd hitting them in the butt to move around. They listened to the music. They followed the light. They somehow were in the right place at the right time. They stood still. They moved. They were like a living organism and individuals. And by being there, they made the connection between we as humans, nature as inanimate, animals as somewhere in between, physicality, ideas. Something that you could only experience by being there and seeing this vision. You could never have explained it to anybody. So art can do that in a way that, frankly, maybe even biology can't, in terms of having us understand, in terms of having us understand something about what matters, asking questions that we can't give simple answers to, and inspiring us to do something about it. So in our group here at the Media Lab, it's called Opera of the Future, but really we're trying to think of how far we can push the experience of music so that it does constantly reveal things about who we are as human beings, reveal something about the world around us, and reveal something about how to make connections with other people. And I want to just give one example. Quickly, I'll say we do make instruments that allow a performer like Yo-Yo Ma to have everything in his interpretation measured, so that by changing the expression of the performance, a cello can turn into a whole orchestra or something bigger than an orchestra. We do this for stages and operas. We make operas with robots, for instance. It's interesting to use technology on stage, but we do that usually to tell a story. In this case, it's a story about immortality and whether technology, in fact, can allow us to live forever. And we talk about it, but we also create it on stage so people can observe it. You should come see the opera, but the answer is technology can't help you live forever. So if you thought it could, it's probably not going to. We do the same thing for try to reach out to everybody, not just Yo-Yo Ma, not just opera audiences, but we make instruments for children, for people of all ages, to do sophisticated things in music, to fall in love with music first and ask their parents for music lessons afterwards. I think that's a better approach than the opposite. And as Deborah mentioned, we do a lot of work to use music to allow people to communicate who wouldn't ordinarily be able to, such as Dan Elsey, who has cerebral palsy, who can't speak, can't control his body, but can use some of our interfaces and software to compose his own music and perform it, like at the TED conference where we went together a few years ago. But just for a few minutes, I wanted to concentrate on this one idea that we're extremely interested in using music to make a radical proposal for how people can break through boundaries, do things together, and understand each other at a deeper level than usual. So for the last couple of years, we've been working on a project called City Symphonies. The idea is that we go into a city, make a portrait of that city using the regular language of music with a symphony orchestra, but also by listening to the city and listening to its sounds that aren't musical, necessarily traditional musical sounds at all, putting those together with music, and we invite everybody in the city to come and make this piece with us together. So everything about it, the shape of the piece, the sounds, how it fits together, the story, the meaning is all something that we make together. So we started this in Toronto, made one for the Edinburgh Festival, the Perth Festival in Australia, the Lucerne Festival, and most recently in Detroit. And the Detroit project was, in many ways, the most powerful because it was a city and a community that really needed a radical means to meet each other and to find common goals. So quickly, for each of these projects, we make an app that allows anybody to go out, record anything in the city. Everything gets automatically tagged geographically, and you can annotate it, so you have this huge library of sounds that builds up, and you can go and explore those. We get all kinds of sounds, so we didn't have sheep, but for instance in Lucerne we had the most musical cows I've ever heard. Really great cows. That cow in the middle was kind of a soloist. We got, this is Henry Ford's very first engine called the Kitchen Engine, which is amazing. It sounds kind of like a chamber music engine. I love this. My symphony actually ended up starting with that sound, with that engine. Street performers everywhere, you know, people who aren't professional musicians but make unbelievable sounds like this guy. So we get thousands and thousands and thousands of sounds. We turn the sounds into little dots. They get analyzed automatically. This allows anybody to go online, listen to sounds, but also to connect the sounds and make their own mixes just by drawing with a mouse. These get traded and morphed and edited massively. We write special software like this, it's called Hyperscore, where kids can write their own music just by using lines and color. You don't need any musical training. This allows, you know, like a whole school system in Toronto, for instance, to participate for kids to make pieces about the city. So, you know, we run workshops all over the place with kids. We work with as many different people as possible, both online, a lot of times in person, like with kids learning hip hop and DJing in Detroit, amateur musicians in Lucerne, improvising musicians and professional musicians everywhere we go. I get hours and hours and hours and hours of this material. It all comes to me at my barn out here in Waltham, and I listen to every single bit of it, things I like best back online, turn it into music with every technique I have on the computer, with a piece of paper. It all ends up as a score and ends up being played by the orchestra. So here's a little tiny 30 seconds of a clip that was made before we premiered. Whoa! There goes Kevin's group. Jeez. Kevin said this was the last presentation he's going to give with this laptop. I'll make sure of that. That's, anyway, here's just a tiny bit about the Detroit piece, and then I'll just play a couple of minutes of it so you can hear it, and then Kevin and I will chat.
I think what Tod's doing with this program, the sounds that we're going to gather and the voices that we're going to hear are new, they're fresh, they're vibrant, and they're full of hope and optimism, which is something that you don't normally see when you talk about Detroit.
I love how he's incorporating like all the sounds of the city, and he's giving people who've lived in Detroit and know the city the best the opportunity to share with the rest of the world what they love about the city themselves.
Detroit is full of people who know how to go out there and get things done, who know how to make a way out of no way.
We're going to make a big piece of music for the Detroit Symphony Orchestra. We're going to show the world what Detroit sounds like. Symphony in D, the piece we made, premiered just a few months ago, and here just are a couple of minutes, little glimpse of the whole thing, and we can talk about it.
If you can't see, we are not done yet. If you don't recognize the genius in refusing to be generic, if you still can't tell the difference between the boom and the bass, You've never been to Detroit. But don't worry, we'll still be here when you decide to learn.
Thank you. So we're always pushing further. I'm giving a course this semester called Music and question mark, where we're trying to look as far as possible in the future and seeing how music intersects with all kinds of things like neuroscience and microbes and space and animals. So in that line of thinking, let's sit down with Kevin and see what similarities we can find.